Welcome to the AI for NetZero webinar series. My name is Ahmed Al-Sheikh. I'm professor of scientific machine learning at Harry-Watt University. Today I will be chairing a session with Dr. Sarah Perez, who will introduce herself.
Hello everyone, I'm Sarah. I'm a postdoctoral research associate at Harry-Watt University. I'm happy to co-chair this session with you, Ahmed.
Thank you, Sarah. As an introduction, the webinar series is designed to bring together experts and innovators from both artificial intelligence and sustainability domains. We have an overarching goal to accelerate progress toward net zero targets, and the series aims to engage and inform communities about groundbreaking research and development at the intersection of AI and sustainability. We received funding from UKRI under 7 different projects under the call AI for Net Zero, and this is our starting points, but we will be expanding the webinar series to welcome contributions from entire research community on that field. Today, we are pleased to have Dr. Otar Akanyeti from the University of Aberystwyth. Sorry about pronouncing the name is a little bit hard for me. Dr. Otar have a BSc in electronic engineering and master's in embedded systems and robotics, and he completed his PhD at the University of Essex, working on formal ways to develop controllers and mobile robots using machine learning and system identification methods. So he has a growing interest in biological aspects of movement and sensing, and he have expanded his research portfolio to human health, mainly focusing on artificial intelligence and chronic disease management. And he's a co-founder of Aberystwyth Stroke Research Group. Without further ado, I would welcome Dr. Otar to deliver his talk today.
Thank you, Professor Sheikh. First of all, thank you for having me, and I'm going to the full mode of my presentation. So before I begin, probably I should tell a few words about myself in terms of as you see my introduction, I've done quite a bit of robotics and a little bit of fish biomechanics and sensing, and recently human health research. So where does today's topic fit in? Just, in science, you never know what endeavors you're going to take. And in Aberystwyth University, we have been organizing an AI summer school for 3 years in a row now. And the idea of the summer school is to invite researchers from different disciplines to bring their own data. And we, bunch of us from computer science, we try to help them to analyze their own data using AI and data science techniques. And through these interactions, I came to know more about how AI can be, and data science as well, can be instrumental in plant science. And through those simple conversations and discussions, and maybe a few sessions of coding, then there was a research project was born from that, and then we got lucky, it got funded. And today, hopefully, I would like to tell you a little bit more about my understanding about how AI and data science can be applied to plant selection and breeding for net zero goal. And the project that I'll be talking about mostly today is Miscanthus-AI, funded by the EPSRC. And the 3 collaborators in this project is Aberystwyth University, University of Southampton, and University of Lincoln. And the principal investigator of the project is Professor John Dunnan from Aberystwyth University. So in a nutshell, the project is all about, can we use AI tools to accelerate the accelerated breeding programs of bioenergy crops? And the idea here is there's already a huge deal of efforts going into improving crops and use them in biomass world. And to do that, quantitative genetics is a prominent and a very established method, and there are a lot of people working on this and just use genetics to identify the potential crops that can improve in terms of yield production, maybe resistance to different diseases and or different climates. And obviously with developing techniques and technologies and genetics, there's a need for multimodal data analysis and data interrogation. And that's where the AI comes in. And this is basically the premise of the Miscanthus-AI project. So what's I was thinking a lot about this, and obviously when you look at traditional fossil fuels, when we use them to generate electricity, it's a one-way street, and we release carbon dioxide into the atmosphere. And the promise of the biomass or plant crops, a subgroup of biomass, is although when you use them to create energy, you can also recapture that carbon dioxide by just growing the plants again. So it's more neutral in that sense in terms of carbon. But what basically separates fossil fuels from biomass is basically the time scale of events, because obviously fossil fuels also is the recapturing carbon from the atmosphere, but over millions of years. But when we talk about biomass, this is reduced to months to years, and that makes it much more feasible and realistic in terms of capturing that carbon in a timely manner. When we look at the energy mix or the energy supply of the UK today, and this is a graph that I produced using data from International Energy Agency, and this graph is based on the data obtained from 2022. We see that we're still heavily depending on natural gas and oil, and the renewable energy and biofuels and waste creating energy is probably constituting more or less 15% of our energy need. And if you take this one step further, if you want to get more information about this and to play around, I highly recommend the website by Kate Morley. And when you go to the website, what you would see is basically breakdown of energy mix in the Great Britain, and you can also see on daily basis of where our energy is coming from or the source of the energy. So when we talk about biomass, there are different ways of generating energy and different sources, and this includes animal fats, manure, vegetable oils, sewage and waste, or agricultural residues. But mainly, and even in the previous chart that I was showing you, that energy comes from wood chips. And that can be slightly controversial, because the ultimate question is where those wood chips come from. And there have been some controversy in the sense that the supplying, if that was coming from forestation of some forest in Canada, and that's basically you are doing some positive impact in the UK, but actually at the expense of some negative impact and environmental impact in somewhere else. And when we look at the percentage, the wood is heavily used, but on the other hand, crops are used much less, plant crops. And I think that's where the Miscanthus-AI project fits in, just to elevate and improve plant crops within the context of biomass and energy creation. So when you start thinking about bioenergy crops, there are many different alternatives, from those that actually we can also consume as food source, to those that actually more like grassy or grass. And when you make a decision about what to plant and where to plant, there are some decisions that you have to do or you have to make and think about, or there are some factors, and one of which is the growth rate. So when you plant the crop, like how fast it grows, obviously the faster it grows, it will have a shorter turnaround, and that might be important to capture carbon sooner, and even in the time scale, that may be a desired property that you want to have on your crop. Similarly, how much carbon you are able to harvest or sequester using the plant is an important parameter. But we also have to think about the alternative use. Like in the example of maize, if you are using it to capture carbon and not as a food source, then you may create a shortage in the market in terms of food, and that might increase the food prices, and you don't want to create another problem while trying to solve one. But we also know that usually these annual plants may also create some damage on the soil, disturb the soil and the water resources, and that's another factor that has to be factored in. And then another one is you want these crops to be able to thrive in different geographic conditions and weather climates, to be able to generally applicable. And it's also important about how efficiently they utilize nutrition from the soil and how much nutrition they need, because that's the input to the system, and obviously those that require less input would be desirable. But we also know that to create energy from these crops, even starting from seeding them in the ground, as well as harvesting them, and also using them in the biofuel plants to generate energy, you may need to use fossil fuel as an input, and that also has to be considered in terms of agroeconomics and especially carbon capture on net zero perspective. And in addition, the economics plays a role as well, because certain plants or crops are much easier to plant and much cheaper to plant than the others, and the economic and also harvesting as well. So the economic aspect of things also plays a huge role, whether these biomass technologies can be realized in real life. So Miscanthus silvergrass is one type of biomass or plant crop that our university has been doing research in the last decade or so, and we like it because it grows tall, up to 10 meters or even sometimes more. So they are very stemmy, and then they have a high in cellularic content. So that's good to capture a lot of carbon. In terms of speed, they also fast-growing plants. So usually it takes them in 1 year you can harvest them, and then that's quite good in terms of in this category plants. And important thing is it has a C4 photosynthetic architecture, which means that during photosynthesis it captures 4 carbons. So in that way, it's superior compared to some other plants that actually capture 3 carbons, which are called C3 plants. They have reduced soil disturbance, and also the way that they transport the carbon from above ground biomass into the roots and the rhizomes is desirable, to just basically in terms of reducing the carbon from the atmosphere. And in addition, when the leaves go on the ground, the leaf litter also is another source of carbon that goes into the soil and helps for the carbon capture. So if you think about how miscanthus plants can be used to create energy, there are alternative ways, and the heat using combustion or generating electricity, or you can also turn it into an ethanol through different processes. And one of the important questions that comes when you think about how biomasses or biomass crops or plant crops can be utilized in the environment, is where we should plant them, and that's an important question, because obviously those lands can be used or utilized for other purposes. For example, if you can grow plants there or food, obviously that's a question that you have to make, because food shortage is a serious concern. It will grow. So one and the most kind of a logical route in that sense is to use the marginal lands. So marginal lands are those that actually are deprecated, so they are not good for food production, but they are not also completely useless. And in those environments, usually planting, and we know that miscanthus and similar crops can survive, and that's an advantage of turning those lands into carbon sequestration farms. And what makes the definition of marginal land is not very straightforward, and there are different ways of doing so. But also, they are marginal because they typically come with different constraints. So it can be weather related, or it can be geophysical, the location, or socio-ecological reasons might push the land to become more marginal, or there might be simply economic constraints, which maybe investing or planting in that land may not be economically viable. But obviously, the reasons that make marginal lands marginal also are important to consider when you're growing your crops in terms of carbon capture, because those crops should be efficient and effective enough to survive in those harsher conditions. And when we look at the life cycle of a miscanthus, so it's an involved process. So you have to plow the land and first, and then you plant the rhizomes, 1.5 a little bit under the ground, and there's an irrigation process, just watering the seeds, and then there's a waiting period, and then you would get the first not even a harvest, but like the growth at the end of the first year is usually low, but then you start getting into the second and the third year, and that's where actually the plant just fully blooms, and by the end of autumn you harvest it, or after winter, and then that's where actually the biomass comes from. And this yearly cycle continues on its own, so you don't have to, that's why it's called perennial plants. So you don't have to do you don't have to provide any inputs, and that's the desired part. And this process can repeat itself for 20 years. So although the initial investment in terms of planting them is a little bit laborious and economically costly, once they are in the ground, they are pretty independent, and then you could just harvest them year after year, and that would give you quite a bit of a carbon capture tool. So when we come to the plant breeding and selection, obviously the idea is to create more optimal miscanthus crops that maybe they grow more, they capture more carbon, or depending on, or maybe they are more disease resistant, or they can survive in different climates. So there can be different optimization criteria or fitness values, but the process is simple. In a very basic terms, you need to select the parent plants that you want to cross. You breed them, you generate the offsprings, you plant the offsprings, and then you let them grow. And in this scenario, you need to, if you're just talking about the lifetime of miscanthus, you have to wait for at least a couple of years to see the full yield. And then you measure the quality of the yield and the phenotype, and then you somehow have to decide whether actually that was better than the existing species. And then if so, you then use that to select the next generation, and then you select the next generation parents, you breed them, you grow them, you measure them, and this process continues until you see a significant improvement in terms of performance. But as you can imagine, this is a slow process, and that's it takes years. It takes years, and that's the reason that accelerated plant breeding programs is becoming more and more desirable, if possible. And the other thing is, it's obviously very there's an expert plant breeders. They have an intuition and a knowledge and a background to see and to identify potential plants to breed in the next generation. And that's a knowledge base, but obviously we don't exactly understand how plant breeders make that decision, like similar to other expert systems. Usually it's not easy to put in words about that hunch, that intuition, and then how you make certain decisions. So it's intuition based, and you can also see it's not unreasonable to assume that there will be some differences between plant breeders depending on experience, knowledge, and the location. So it's a very detailed, it's a very traditional process, but it has some limitations. And accelerated breeding programs would like to complement this approach and provide another tool to plant breeders. So it's not about replacing plant breeders, but it's almost like providing them a little bit more insight and information at the genomic level. And then if we can predict based on the genomic material or the genomic DNA of the plants and how it will interact with the environment, and if we can look at that genomic environment interaction models to predict the future phenotypic traits, then maybe we don't have to wait until these plants fully grow, and we can reduce maybe instead of making selections every 3 years, maybe we can start making those selections in every 6 months based on their early growth phenotypic traits, combined with the genomic and environmental information. And if so, we can go through more generations in a reduced time. So when it comes to data, obviously there are different sources of data, and simply, if you want to monitor the environment where the plants grow, this could be the soil. So you can measure the moisture in the soil, the water levels, porosity, and alkaline or different chemical properties of the soil, and that might provide useful information in terms of plants' performance. Similarly, weather plays a huge role. So you can have the same species, same genetic material, but depending on the environment that they grow in, they might produce very different biomass. And so that has to be included in the decision-making mechanism somehow. But in addition, there's a lot of imaging and non-intrusive imaging going on at different levels. So this could be simply flying an UAV or drones over the fields and capturing some spectral imaging, or you might have a handheld camera and just go into the field and take a couple of photos, and that might give you, and you may want to process that information to estimate the stage as well as the amount of yield that specific plant will produce. But there are also some more involved measurements, so you can, which may include taking a ruler or a measuring stick and going into the field and measuring the stem of the plants, as well as the height, You can count the number of leaves, the diameter of the leaves, and so on and so forth. And then you can also actually collect some of those parts. You can take samples from different plants, and then you can actually look at the chemical decomposition to measure their cellulose and lignin contents, which are key when it comes to energy production. Or you can go in further down at the chain, and you can actually start looking at the genomics, basically DNA or transcriptomics or proteomics, or even metabolites, to actually get the bioinformatics information. And recently, near-infrared spectroscopy is becoming more popular to look at the biochemical properties of the plants, plant samples. So what's interesting about this data is they come from different sources, and they're also very heterogeneous in the sense that their resolution is different, their time of collection is different, this temporal and spatial resolution is different, the magnitude change quite a bit. Some of them are done once, some of them are done on a periodic manner. So for example, if you're measuring the stem diameter, you can do it simply every week or every 2 weeks. On the other hand, if you're sampling data and looking at the which proteins are turned or which genes are turned on and off, that's very time dependent, and so every time you take a sample, you will have a different profile. And obviously the efforts and the costs that are associated with each data type also changes quite a bit. So just to highlight, when you're measuring the physical traits of the plants, you can look at when they emerge from the ground for the first time, and that is a useful predictor about their future performance. You can also look at the stem diameter of each branch, or the canopy height, or when leaf senescence, so when the leaf starts aging. But also you can actually, once you harvest the plant, and then you can actually dry them in special chambers to remove the water. And then when you measure the when you measure the weight of the dry matter, that will give you, for example, a yield. And that's like a one-time measurement at the end of the harvest. And similarly, you can look at the SNPs, basically the differences between offsprings in terms of in the nucleotides, or you can actually look at the RNA level, or the protein level, or at the metabolite level. So coming back to the Miscanthus-AI, what we are interested in is a multimodal data analysis for automatic phenotype trait analysis, and that includes in the work package 1 using robots, UAVs, cameras. And in work package 2, we try to basically improve the genomic selection, and in this case we are combining we aim to combine data from multi-omics, nurse, and early phenotypic traits data. And in work package 3, which I'll finish my presentation on, is more about getting into the brain of the plant breeders and trying to see what are the key variables or the key processes that their decisions are based on. Because if we could understand the way that they decide which plants are good to produce in the next generation and to use in the next to produce the offsprings of the next generation, might provide a very valuable insight about the decision process of plant selection and breeding. And our goal in this project is to create explainable rules, plant breeding rules. So in a sense, you can argue that we are after a digital plant breeder to elucidate that decision-making mechanism. So it's extremely important. So this photo comes from our National Plant Phenomics Center at Aberystwyth University, and even here you can see the young Miscanthus plants. And what we want to do is, rather than trying to go each one of these plants and make manual measurements, like, can you just take a single photo, for example, and then try to extract key information about the phenotypical traits. And as you see, analysis of this type of images is not very straightforward. There's a lot of occlusion, there's a lot of overlap, the color is more or less the same, and it's kind of a very good example of where AI can come in and with a little bit of a help and training, try to infer some knowledge automatically in these non-interest destructive imaging data modalities. And when we talk about non-destructive imaging here, the alternative or the traditional method would be to cut some of these plants, or to take tissues from some of these plants, to lay them flat, and then look at them under microscope and do those counts or measurements manually. So in work package 2, like I mentioned before, the ultimate goal is to combine environmental data, the early growth properties of the plant itself, some nurse data, and also the genomic data, and try to predict how this plant will look in 3 years or 2 years' time. But again, if we can do so, and if we can do this accurately, what that means is actually that we don't have to wait until the plant grows. We can start deciding whether we want to use them as parent or not for the next generation at a much earlier stage. And if you look at the nurse data with a little bit closer, what you see is the way that you collect nurse data, and which is basically what I'll show in the following few slides. So before we try to combine information from these different sources, we first decided to look at what the potential is for each one of them independently. Because if you know that, for example, nurse data can allow you to predict the phenotypic traits by X amount of percent, and then you can start evaluating, okay, so what if I combine nurse data with the SNPs data? Can I increase that 0.X% to 0.X plus 5%? And I think those type of baseline studies will help us to set the bar and see how much we can go about it. But just to go a little bit more in detail, so this is a typical nurse data that you would get from multiple genomes, so multiple individuals. And as you can see, that actually the data is highly correlated. So they always follow a very similar shape, with maybe some differences in amplitude and some pattern, or the profiles may vary very subtly. And extracting information from this is not straightforward, and we have been doing quite a bit of a data standardization and transformation to enhance the differences between individuals. But the way the data is collected is you get a sample from the plant, you chop it at a fine, almost like a dust level, and then you start shining light at different wavelengths, and you measure the reflectance, and that gives you a profile across different wavelengths. So usually it starts around 400 nanometer up to 2400 nanometer, and in that range you get each plant or each individual has a profile, and that based on the profile, the question is, can we predict the future plant growth performance? And to do that, after we transform the nurse data, we decided to start with a classical statistical / machine learning approach, without going into too much without using too sophisticated models, again, to see what we can achieve to begin with. And so what we do is, if you look at here, you're talking about at least almost like 2,000 readings per plant. And in our data set, as I'll show you in the next slide, is we have around 100 136 individuals, and there are repetitions, 4 repetitions. So our it's a very sparse data in the sense that we have less data points than actually the features. So in this type of situations, somehow reducing the number of features or transforming the data to get a smaller, creating a smaller feature set or predictor set is desired. And to do that, we decided to start with the principal component analysis. And then we saw that actually the first 3 to 5 principal components were able to explain the 99% of the variation in the nurse data. And that's why we decided to use the first 5 components and see what we can achieve. And then we fit these 5 components, high rank components, into a multilinear regressor, and then look at the yield or the dry matter prediction. And the results look promising. So we were able to, our coefficient of variation is around 0.31. And when you look at this, it doesn't look super great, but it's a good starting point. And these experiments have been done 10-fold cross-validation. So we basically kept 90% of the data for training the model, and then predict the remaining 10% and look at the performance. And we repeated this process 10 times by reshuffling the training and test sets. And we saw that our range can go up to 0.2 to 0.4 in terms of prediction accuracy. And this performance is not worse than genomic predictions so far. So if you repeat this process using the SNPs data, you would get a similar performance, suggesting that the nurse data is worth to look into more. And our next step from this point would be to see whether actually we can take these numbers higher if we start combining data from SNPs and nurse. And obviously, the idea there is we believe that they give us slightly complementary information, and that should help us to improve the prediction performance. So just to give you a few more details about this data set. So this is a data set that generated the Aberystwyth University, and the species that has been used is Miscanthus-AI, and it had 136 genomes, so offsprings, and then we have data for SNPs and nurse, and then we also measured 14 phenotypic traits, including the yield that I showed you in the previous slide. But there are also some other traits that might be useful to predict, including lignin and cellulose for energy harvesting perspective. So when it comes to work package tree, so work package tree is more about interviewing the plant breeders and giving them some questionnaires to fill in, and they try to understand how they go about plant breeding business. And here you see like two questions or example questions that the plant breeders should fill in. And what we try to do is we try to accumulate or collate that information and try to use logic and reasoning and computational argumentation to derive some rules that explain the way that plant breeders go about their business. And this is going on, so we are at the interview stage now. But this also opened up another interesting project that we have started thinking about, and this is partly related to my background because I am more interested in neuroscience and looking at decision making. So, and we have an eye tracker, and basically the one you see on the right, Toby glasses. So what this does is it has an external view camera, scene camera in the middle. It also has 8 illuminators per lens and 2 cameras per lens that actually look at the pupil. And doing so, the glass is able to track the gaze patterns, so where people are looking at in the scene, and superimpose them onto the image are taken from the scene camera. So in this way, when you're walking freely around, or when you're basically sitting in front of a desk and looking at photos of different plants, you should be able to see the eye patterns or the gaze patterns of the plant breeders about what cues or visual cues they are looking at when they're evaluating the quality of the plant. Similarly, we can also use NFNears, which is basically a brain imaging technique to record brain activity at the cortex level. And this is also very exciting because you can also start looking at brain activity when breeders are doing always making decisions about the depending on the scenario. This could be just walking in the field, or it can be like in front of a computer. And by looking at that, we can see actually when the brain gets activated, which parts of the brain are activated, and how that fits into the decision making. And the reason that we are interested in this is because we believe that like other fields, especially the expert decisions, there's a possibility that they are made subconsciously, without actually realizing or controlling actually how those decisions are being made. Although when you ask the breeders, they will give you a very reasonable explanation about how they make the decision. But in fact, they might not be that might not be the mechanism that they are using to make the decision. So the decision making might be subconscious. With this approach, we are hoping to capture that subconscious part of the decision making. So it might provide us a slightly different view about like how they evaluate the plant quality for future, to decide whether actually they should keep them in the pool or not. And just to demonstrate you the strength of this approach is, I'm just going to start talking about something, a completely different project that we are currently running in Istanbul. So I'm in Istanbul right now during this presentation, and what we are doing is we are looking at in one of the busiest areas in Istanbul, 20 million city with a very also like a very crowded place. So how humans navigate in crowded spaces. And here is a very simple croquis. So we have a route that we ask participants to follow. Imagine you start from S and then you finish at F. It's more or less straight in a crowded downtown area in Üsküdar. And there are a lot of people coming on your way, and you cannot predict. It's very difficult because people walk in very different directions. There are people coming off the bus, there are people walking on the street, there are people crossing the streets, and it's not easy. It's a very dynamic, unpredictable, and surprising environment. And our goal is basically to look at how participants decide to either go through the crowds versus go around them while they're going from start to finish. And we ask them to wear the Tobii glasses, and also we measure their behavior using IMU accelerometer data. And here is an example about our approach. So what you see is on the left is a typical scene from those environments. As you see, many people. So this is a participant looking into the world, and as you can see, there are many people coming oncoming flow. And on the right, on the middle image, you see the fixation. So where the participant is looking in that particular frame. So where their gaze focused. So our goal is to see whether actually we can create a machine learning or AI model that actually can take the raw image and predict the gaze patterns of the participants. So if we can do so, that would give us the indication that actually the way we process the visual scene is predictable, and it's also deterministic. And there are key feature or visual cues in that scene that makes we base our decisions on. And then what we'll do is we can then use, once we know exactly where a person is looking at, and then you can apply a spatial filter to create more like a human vision, where we have a foveal, where actually high acuity region versus peripheral vision. And that can be fed into the second AI model to predict the action that a person is taking, whether actually to turn right or to turn left or go through the crowd. And the same architecture can be easily applied to plant breeding scenario. So again, the first step is to predict, what visual cues a person is focusing on while evaluating the quality of the plant. And the second model should basically use those regions or the information from those regions to then predict, okay, whether actually that plant is good enough to keep it in the next to create the next generation offsprings or not. And here's a video of our prediction model. So here you can see me. So this is just person. Our participant is walking around, and the blue shows you the actual gaze patterns of the participant, and the red is our predictions. And as you see, there are sometimes differences, but the majority of the time there's a good overlap between red and blue circle, suggesting that we are, or at least it basically gives us a hope that actually we should be able to track where a person is looking at reliably, and also predict where they will look at based on the frames that are coming on. And what you can do is then you can actually combine this information with the pause estimation algorithm that have been previously developed. So in this case, you can track the people that come in on your way, and if you can do that, you can also start combining people information or the position of the people with your gaze patterns, and you can see whether how much of the time you're looking at people's heads, how much of the time you are spending looking at people, versus how much of your time you're looking at maybe some environment on the floor, on the what's going around you. And that's important again within the context of plant breeding. You can use this, for example, as experts, we might argue that we typically measure, for example, stem diameter,
It seems that we have lost a speaker.
then it will support our hypothesis. Otherwise, there might be some additional visual cues that we haven't thought about before that might be actually informative when it comes to biomass yield prediction. So here, from that, you can start extracting information, like on the top columns, you can see different participants walking, and on the rows, you see the instructions that they receive. So when you see food, that means that we ask them to look at the food, and when that happens, you see that the percentages that we spend looking at food is not as much as the environment, but it's there. But if we ask them now to look at trunk, then we see that less of a food focus, but more of a trunk. So there's a transition, almost like a 10% transition in the gaze patterns. And we also, if you ask them to walk freely, we see that some people spend more time looking at naturally to food, and some people spend more time looking at the trunk. So in this condition, we get the more natural behavior or tendency about what people do. So this is exciting, like I mentioned before, because I believe that the same methodology can be used in plant selection and breeding. to reveal novel insights about decision making by the plant breeders. And with this, I would like to conclude my talk, and thank you for listening, and happy to answer your questions.
Thank you, Otar, for the very exciting and extensive talk. I would like to remind the people who are attending, please post your questions in the chat. We have now started the Q&A session, and I'm glad to see that we have early questions. One of the questions I will show here, it's how much do we know about the impact of growing Miscanthus in the UK for biodiversity? And the question, are there any positive or negative secondary impacts? Which is alludes to any action we do to the environment, there might be some unintended consequences.
So I'm not an expert on this, and I don't want to speculate. I'm so sorry, but what I know with little my information, I know that it's good for worms, and it's less. Miscanthus is less. It's not disturbing the soil as much, but that's the extent of what I know. So I'm sorry, but yeah.
No, fair enough. And a question from my side here as a follow-up. What's the scale we are talking about? What kind of land coverage in the entire UK? Are we talking about 1% or 10% of the UK land coverage? Is there any rough guides?
Yeah, so yeah, I don't know exactly how much land we have to invest because it comes to I start looking into that. I haven't come to the conclusion yet, but what I see is there are numbers in terms of BTU, how much energy you can create per pound or per half a kilo, and how much biomass you can create per hectare. So those numbers are out there, and those numbers can be used to make those calculations. But when it comes to what we need is then the definition of marginal land becomes really important here. I think the economic side of things is the bottleneck, because it's always a question of, okay, do I use this land for Miscanthus or do I use it for something else? And when it comes to that, it's not just about how much energy you can extract from that land, but it also how much money and resources you need to invest to get the plant growing there. For example, if it's on the highlands of like Wales, for example, we have lots of marginal lands on the hills. So getting the plow machine on those hills to seed the plants itself is a cost. And I think so I don't have exact numbers, or I don't have a clear answer, but I know what I'm trying to tell you. There are a lot of factors that have to play in to provide those numbers effectively.
There is a second question here about the intuition of plant breeders, and we assume that they are good first, and they play an important role in the selection process. Considering this, the several plant breeders will take different decisions, and considering this variability, how scalable is AI-guided breeding approach for Miscanthus? And there is a follow-up part of it because it's being trimmed, which is here. Could this method be easily adapted for other plant species and agricultural contexts?
Yeah, so let me start with the second. I think so. Once obviously every plant. So what happens here is, what we need to understand is, first of all, the genomic profile of the plants may vary, and the complexity also may vary. So doing it one plant doesn't guarantee that you will be able to translate that directly to another plant. So that's one thing, but at least it gives you, in principle, it should be, in terms of there's a at least the method is valid, but it's all about finding the correct QTLs or the right areas within the genome that actually we need to target, because we know we believe that, okay, so the performance of the plant should come from the interaction between its genetic and environment. So these are the two variables. We don't think there's another unknown factor playing. So it is a deterministic system, but it's super complex, that actually behaves unpredictably because we don't understand. If we had the theory, if we had the full genetics, and if we had the environmental information and the capacity to process that, we should be able to solve this problem. But it's always a matter of like probably other disciplines, getting there is almost impossible because of the nature of the complexity. And that also applies to plant breeders, and it's a good point. So it depends on there's going to be variation there. So that's why we need a human in the loop process. So it doesn't have to be, it's not about us blindly copying their decision or trying to mimic their decision, but trying to create a reasonable and logical argumentation that helps us to guide that process or accelerate that process. So when we extract these rules, those rules have to be moderated and also have been discussed and maybe made publicly available, so people can actually feedback their opinion. And then it has to be like an ongoing iterative process to bring it to a stage that actually can be used in the real practice.
Fair enough. I think one more question before we run out of time is the general field of explainable AI. What's your view about explaining? I know that you touched that about taking roles and then judging these roles, and but in most times we don't have roles. We have a black box. So we have a big neural network. We don't know what's going on there.
Yeah, so that's very true. I think it then comes onto the sub problems that we are using in this domain. So, for example, if you're trying to count the number of stems in an image, obviously your error margin, there's going to be an error margin, but maybe that error margin, or you may not be super interested in understanding that decision it's per se. On the other hand, if it's more about deciding which plant to use and which plant don't use, obviously you may need more explainability there to be able to relate what features of that plant that make you make that decision, or make you feel like this is a more breedable plant than the other. And I think when it comes to that level of stages where actually decisions really make impact in terms of deciding whether to keep or not to keep a plant, we would need those more like maybe rule-based and more, transparent decision mechanisms, so that actually we can relate back and forth with the computers. But maybe at the very low level, when we are processing those images to extract those features, we may not need that experience. And in fact, what I'm seeing is it's the same. For example, the gaze patterns that I show you suggest that we are also doing a lot of as a person, I can explain the logic of my decision making, but I think what we underestimate is my logical explanation may not be the true reflection of what went behind the scenes. And I think these gaze patterns is a good proving of that, because for example, there have been a study in terms of interview with pilots about how you fly the planes, and they were giving a lot of arguments about, okay, if I see this, I do that, and etc. But then when they look at their gaze patterns, they find that what they believe that they are looking at is not where they are looking at. So there's this level of subconscious decision making that even is not transparent to us, and we cannot explain straightforward. So it's a problem, and I don't think it will go away. especially these models are becoming bigger, especially AI models. But I think what we need is responsible scientists that actually don't sell them as magic. Like in our case, we have 100 data points and 2,000 features. It's a sparse data problem, and if I train a network with million parameters, I know that it will overfit and it's not going to provide anything useful. And I think those things have to be requires good practice about when to use them and how to use them. Sorry, I'm giving very vague answers because these are very good questions, but also the answers are not very simple.
No, we really appreciate the answers and you are providing, and we know that these are very challenging questions, and we as a community we should be upfront. The magic does not exist, and we need to be proceed carefully because the decisions have impact. So I think we ran out of time by 1 minute, but I would like to thank our speaker again, Professor Otar. The talk today was very informative, and I learned a lot myself from this talk. So thank you for sharing your thoughts, and for all our the people who are attending. Thank you for joining us, and please support the webinar series by spreading the word, by participating, and even volunteering to speak. Next week, we will have 2 talks by early career researchers. So please come and support our early academics. Thank you again.